Okay, hello everyone, and welcome back to the Introduction to Bioinformatics online course. We're continuing today with Module 1, and in this session we'll be having a look at protein classification and protein resources. So by the end of the lecture, we hope that you'll be able to understand how protein sequences are annotated, to understand the different levels of protein classification, and identify the key resources used for classifying protein sequences. By the end of the practical session, we hope that you'll be able to differentiate between the different protein classification methods, to be able to use the appropriate tools to annotate a protein sequence of interest, and to be able to access and retrieve information of interest from these protein resources. So just to recap before we continue about the types of data we've had a look at so far. So in session 1, we had a look at some bioinformatics databases and resources. And in particular, we had a look at, in the practical session, at accessing information on nucleotide sequences, together with the associated annotation for those nucleotide sequences. And that included genomic DNA sequences, as well as mRNA sequences. And in this session, we're going to be having a look at, in more detail at the protein sequence databases and resources, as well as how those sequences are annotated. So if you have a look online, again, you'll find that there's a variety and a large amount of different protein resources, as well as protein databases. And again, just to reiterate, it's quite important to differentiate between a resource or website and a database. So if you can think about the databases are the tools, the databases are where we store our raw sequence data or raw information. And then on top of these databases, we have different websites and resources that have been built to allow us to easily access information from those databases. In most instances, the resources that we'll be having a look at will allow us to access information from various different databases at the same time. So that is one of the advantages of using these resources, is that instead of having to go to each one of these databases individually, we have these great resources that allow us to access information from multiple databases at the same time and provide a good visual output in order to analyze the data that we get back. So it's important to just keep that in mind to differentiate between databases and resources. So to begin, let's have a look at some of the protein databases that we know about. So in session 1, we had a look at NCBI protein database, or we had a look at NCBI, and you might have come across the protein database. So this database contains protein sequences from GenBank, some manually curated protein sequences that are generated by RefSeq, as well as records from some new resources that we will speak about in a bit of detail in this session, SwissProt, the protein information resources, and PRF and PDB. We also then have a protein resource at the EBI. So this is actually a resource, and it's known as the Protein Knowledge Database. So this resource contains a comprehensive set of protein sequences, together with rich functional annotation information, which is accurate and consistent, and it'll give you any information about a particular protein sequence of interest that you want to find more about. UniProt KB is essentially very widely used, and essentially I think it is the best resource for finding any protein annotation information. The knowledge database is divided into two main parts, so it's SwissProt and Tremble. And this is a diagram just showing you the flow of information, and this is in particular for UniProt KB. So at the bottom, you'll see that these are the raw sequence databases, which they've termed external resources. So that's EMBL, GenBank, and PDBJ. So that's where raw sequence data is submitted by anyone who generates it. In particular, now we're only talking about protein sequences. So that data from the raw external resources is piped into a resource called UniPark. And this is essentially just an archiving system that EBI uses. So all information is stored and archived and reviewed in this UniPark database. The sequence data from UniPark then goes into the UniPark knowledge database. And again, I said there were two parts. The first part is Tremble. So Tremble actually stands for Translation of EMBL. So translation of EMBL is all the sequences that are submitted to the, all the nucleotide sequences that are submitted to EMBL are automatically translated into protein sequences. And those are all stored in the Tremble database. Okay. So the first thing you must realize is that there isn't experimental evidence for a protein existing, for all proteins that are in Tremble existing, because those are just nucleotide sequences that are automatically translated into protein sequences. What happens is sequences from Tremble are then undergo a process of manual curation and annotation, and this is where the experimental evidence is then linked to the protein sequences. So if there is experimental evidence for a protein sequence existing, that is then linked to the protein sequence, manually curated and reviewed, and that then becomes an entry in SwissProt. Okay. So you can appreciate the fact now that any entry that you see in SwissProt is generally of high quality, and you know that there is a level of experimental evidence supporting the existence of that protein sequence if it is in SwissProt. Okay, so that's just very important to keep in mind. Then there's a couple of other databases. So there's other resources. So there's a proteomes resource, which are just protein sets expressed by different organisms and categorized by different organisms. And then there's something called UniRef, which we're not going to talk about in too much detail, but essentially it's a resource where they have tried to cluster different protein sequences based on different levels of similarity. And this is used for some of the annotations that is done by the annotations that are generated in the UniProt database. Okay, so let's have a look at some important concepts for protein classification before we move on. So we can classify proteins based on two main types of approaches. So it's based on sequence similarity or structural similarity. And in most cases, it actually is a combination of both of these that is used to classify and annotate proteins. And it's based on the idea that we already have, in most instances, we have groups of well annotated protein sequences of which the function of those protein sequences is known. And we can use that information to then try and elucidate the function of proteins for which we don't know what the function is, or for novel protein sequences. So proteins can be classified in various different ways. So they can be classified into different groups based on the families to which they belong, and this is based on sequence similarity. They can be classified based on the domains, the functional domains that they contain, and they can be classified based on the sequence features that they possess. In most instances, there's an overlap between these three different methods, and hopefully that will become clear to you as we go through the lecture. And again, the basis for this is that protein families share a common evolutionary origin, and based on their related functions and similarities in sequence or structure. So this is just a hypothetical representation of a hierarchy that is used in protein classification. So we have different levels, and these different levels are defined based on the similarity in sequence as well as the similarity in function between different proteins. So let's just unpack this a bit more. So at the superfamily level, we usually have, this would be a large group of distantly related proteins. So they might not share a high amount of sequence similarity, however they may share a few common functions. So these might be a larger group of proteins that share common functionality in terms of the functions that they carry out. Those can be further broken down into the family level. So these would then be groups of proteins that might share more characteristics. So they might have a higher level of sequence similarity, and they may contain particular functional domains that are unique to that particular group of proteins, and that would then differentiate them into a particular family based on the function of those domains, or repeats, or sequence features. And then we can further break those down into subfamilies, and these would be groups of proteins that are very closely related to each other and share a large amount of sequence similarity, share a large amount of functional domains, and are closely grouped together based on the functions that they carry out in the body or in the system that they're involved in. Okay, so that is the one level of classification based on sequence similarity and the identification of functional domains. So now I want to go on and speak a bit about what are these different functional elements that we are able to identify within protein sequences, and how can we use that to be able to further classify and annotate protein sequences. So we'll go into that in the next lecture.